Florian, over to you to take hold of the screen, and I'll duck out, and then we'll just take questions at the end of this all. Thank you all very much for coming. Florian.
Yeah, thanks for the introduction, and thanks for inviting me. I'm going to share my screen here. And as you can see, I have a very general title, but we'll go to a very special place with this today. And I'm mostly going to share data that is not published yet. And let me know if you can see my slides or if you can't hear me. Just to start out with a very brief introduction. Of course, when SARS-CoV-2 emerged, many of us are from outside the corona field. We looked at the corona literature, we looked at the sequence, we looked at the structure of the virus, and it was clear to most of us that we need to look at the immune responses to the spike protein if we want to look at protection, because the spike protein, of course, is the protein that attaches the virus to our cells. And that makes sense because, of course, if you prevent that attachment, you neutralize the virus. And if you neutralize the virus, you get protection, right? So very simple concept. You have your virus that can infect cells. And if you have neutralizing antibodies, you stop that infection. It's very clear. A lot of correlates of protection are based on neutralizing antibodies, and it made sense to look at that. And a lot of the efforts initially to elucidate these mechanisms of neutralization were focused on the spike protein, and specifically the receptor binding domain of the spike, which is shown here in red. But where I want to go today is more in the direction of non-neutralizing antibodies that bind to other parts of the spike protein. And first, I'm going to talk about a project that we have already published, but you'll see where this goes, specifically with the monoclonal antibodies that we got from this project. And the person who is really spearheading this in my lab is a postdoctoral fellow, Jordan Clark, who is actually from the UK and who joined my lab during the pandemic. And so initially, when we got vaccination started here at Mount Sinai, this was in December of 2020, we took our first 6 participants in our study that got vaccinated, and we wanted to have a closer look at their immune responses, both from a serological perspective, but also from a B-cell perspective, from a monoclonal antibody perspective. And so this is shown here. Of course, we had already a lot of study participants in our studies who had natural infections. And we have characterized their immune responses, or we had characterized their immune responses at this point in time very thoroughly. And we had our 6 first vaccinees, and we had serum samples from those people as well. And so we wanted to compare that, and that's what you see here in A. These panels always have 2 parts. On the left side, you see convalescent individuals who were previously infected. We stratified them into low, intermediate, and high responders. It's just one time point post-infection, basically. And here on the right side, you always see our 6 initial vaccinees. And here we have a little bit more in terms of time points. So what we can see here in A, when we look at the spike binding response, so this is against full-length spike in an ELISA, we see that the vaccinated individuals, at least 5 out of the 6, have a spike response that's higher even than the people who are strong responders after infection. When we look at RBD, this is shown here on the right side, we see basically the same. Again, we have variable responses after infection, but 5 out of the 6 vaccinated individuals have a very strong response to RBD, better than what you see after infection. What was interesting here is that the kinetics are a little bit different. The spike titers are more stable than the RBD titers, which fall off over time. That is interesting because RBD is, of course, part of the spike. And we're investigating this now in a larger group. Of course, we also looked at neutralization. And while neutralizing activity does correlate relatively well with binding activity, it doesn't correlate perfectly. So you have people who have low binding activity, but relatively strong neutralization. And what we also saw was that, yes, after mRNA vaccination, you do get strong neutralizing activity, but maybe it's not that much stronger than what we see in the high convalescent group. And because we saw much higher RBD titers and spike titers, we started to look at ratios. So we calculated ratios between spike binding activity and then neutralizing activity. This is shown here for the convalescent individuals. And what you can see here is that the ratio is variable. The people who have low titers have the best ratio. So the lower the number here, the higher the ratio of neutralizing versus non-neutralizing antibodies. People with higher titers have less favorable ratio. And with the mRNA vaccinated individuals, we have the I would say worst ratio. Of course, the absolute titers are high, they have good protection, but they have much more binding antibody than they have neutralizing antibodies. And please keep that in mind for when we look at the monoclonal antibodies. We also wanted to look at backboosting. And as you know, humans are infected seasonally with 4 different human coronaviruses, 2 of them alpha coronaviruses, 229E and NL63, and the 2 beta coronaviruses, OC43 and the HKU1. And we wanted to see what happens after vaccination to our responses to these seasonal coronavirus spikes. And it turned out there's not much that happens with the alpha coronavirus spikes, but there is an increase after SARS-CoV-2 vaccination in reactivity to the OC43 and HKU1 spike. So we see some back boosting here. And this also makes sense because alpha coronaviruses spikes are further away from the SARS-CoV-2 spike than the beta coronavirus spikes, because SARS-CoV-2 is of course also a beta coronavirus. So that's interesting. And again, keep that in mind for the monoclonal data. Now, we wanted to look at the B-cell response here as well. And we didn't want to bait with RBD, for example, to pull out reactive B cells. People had done this before, and of course, they found a lot of neutralizing antibodies. But we wanted to look at in a more unbiased way. And the way to do that is to sort plasmablasts, which can be sorted based on phenotype. And they are plasmablasts are a transient population after vaccination in the periphery. They peak around day 7 and then they disappear. But during that time, you can fish them out, and most of them are going to be antigen specific. I just put a little schematic here. This is very, very simplistic. Of course, this is much more complicated in reality. But basically, when you respond to an antigen, the B-cell response can either come from naive B cells, or if you have seen a version of this antigen before, the same antigen from a memory B-cell pool. And the first response that you get are these plasmablasts, right? You get a lot of them. They produce a lot of antibody. They are in the periphery, but they basically die after 15 days, they're gone. But they make the first wave of antibodies. And of course, later you get long-lived plasma cells, which also migrate to the bone marrow and more memory B cells. But the nice thing is, as I said, you can sort these plasmablasts phenotypically. You don't have to bait them with an antigen, and that allows you to have an unbiased look. And so we did that together with Ali Betty at Washington University. And from 3 of these 6 subjects, we were able to get a bunch of monoclonal antibodies. They are shown here on the left side. So we got quite a few from subject 3 and subject 6, fewer from subject 5. But when we express the antibodies from these plasmablasts, we see that to various degrees, they bind to the SARS-CoV-2 spike. We were interested to see to what part of the spike they bind. And so we probed recombinant RBD, NTD, and S2. And we see that about a third of them bind to RBD, about a third bind to NTD, and about a third bind to S2. There were actually, I think, 3 antibodies in addition that didn't bind to any of the components, but bound to the spike. And we're still trying to figure out to which part of the the spike they are binding and what their epitopes are. But most of them map to one of these three pieces. And of course, when we had all these antibodies, the next question was, are they functional, right? And so we ran a neutralization assay. In our case, that's a microneut that's run at biosafety level 3 with the actual virus. And to our surprise, very few of these antibodies were actually neutralizing. For subject 3, we had one neutralizer, which targeted the RBD. For subject 5, we had an NTD binder that neutralized. That was actually the strongest neutralizer that we found here. And in subject 6, we had 4 NTD and 1 RBD binders that neutralized. But very few neutralizers, right? There's a lot of antibodies that bind, but that do not neutralize. And that goes well with what we see in serology. And then we also wanted to look at reactivity to these seasonal coronavirus spikes, and we didn't find any reactivity to alpha coronavirus spikes, but we found that 5 antibodies to variable degrees bound to OC43 spike, and 3 of them bound to the HKU1 spike. So there's cross-reactivity. Most of these antibodies target the S2. Again, we haven't mapped the specific epitopes yet, but it makes sense that they target the S2 because that's more conserved. When we then started to look at these antibodies in the context of variants, we saw that the neutralizing antibodies, which are here, please ignore those. These are mouse antibodies that we tested at the same time. We can see that they neutralize, as we have shown, Washington 1 or wild type SARS-CoV-2 well, but they have basically no neutralizing activity against the Omicron variants, and against other variants, very little neutralizing activity. However, if you look at binding, and this is shown here, split up into the RBD, NTD, and S2 antibodies, we see that most of them actually maintain binding. Even the neutralizing antibodies that drop off in terms of neutralization, they can still bind to the spike protein. And that's something interesting that we've seen before with influenza, that you can lose neutralizing activity because the affinity might not be high enough anymore, but the antibodies still bind. And the non-neutralizing antibodies turned out to be mostly broad binders as well. And so we had a lot of these non-neutralizing antibodies. And of course, the question is, what do they do, right? So you get them through vaccination and then they're there. Do they have any benefit? And we tried to test that with a very simple mouse model. So we transferred the antibodies into biopsy mice IP at 10 mg per kg 2 hours before they were then challenged with a mouse-adapted SARS-CoV-2 that we got from BI Resources. Ralph Baric had deposited it there. And so this virus is deadly in biopsy mice, and then you can record weight loss and survival after challenge. And so we did that, and sorry about the confusing graphs here. They're unfortunately not in shape yet. But the message that what we saw here is that no matter if you looked at RBD antibodies, NTD antibodies, or as you will see in the next slide, S2 antibodies, we of course saw protection, and this is indicated here on the right side, from mortality and also from weight loss with the neutralizing antibodies. And of course, you would expect that, right? But for many of the non-neutralizing antibodies, we also saw 100% protection. And that was remarkable and not expected. As I said, we also see this with S2. So none of these S2 antibodies were actually neutralizing, but some of them had pretty good protective effect, specifically. Sorry about the background noise. Specifically here also this antibody, which protected very well from weight loss. And one of the antibodies binding to the unknown epitope also protected relatively well from weight loss. Now, they are not neutralizing, and the question was, maybe we just see an immune modulatory effect, and somehow the mice have a different cytokine profile. The virus might not be controlled because they're not neutralizing, but they survive somehow. So we wanted to look at control of virus replication, and this is shown here. Again, what you see in the upper panel are lung titers from these mice at 3 days post-challenge. for the S2 antibodies here on the left, the RBD antibodies here, the anti-D antibodies, and then the antibodies with the one antibody that showed protection with the unknown epitope. And in the lower panel, you see what happens 5 days post-challenge. And so what you can see when you compare this to the negative control here, these are mice that received a flu antibody. You can see that there is a strong reduction, even 3 days post-challenge in virus titers in the lung. And in many cases, 5 days post-infection, the virus was cleared already. And so the question really is what the mechanism is here. And of course, we know from influenza, from HIV, and also from filovirus infections that the Fc part of the antibody can play a big role in protection. And so two of the mechanisms that can play a role here are either ADCC, so basically engagement of NK cells through the Fc receptor that's bound to infected cells, and then the NK cells clear the infected cell and you limit virus spread, or ADCP, where macrophages or phagocytic cells are engaged that then might also clear infected cells. And so we wanted to measure that, and for that we use a reporter assay, actually based on a commercial solution that Promega offers, where you have reporter cells that express different Fc receptors, and if they're engaged, you get luciferase expression, and you can measure that. And we set up a system to do that at biosafety level 2. We have Vero E6 cells that we infect with Newcastle disease virus vector that expresses a lot of the hexapro spike protein. We incubate these cells then with the monoclonal antibodies. We add the effector cells, and then we can see activity. And so when we do that, and this is shown here again, split up for the different types of monoclonal antibodies, you can see that many of these monoclonal antibodies, not just the non-neutralizing antibodies, show activity in an ADCC reporter assay. Some of them also show activity in the ADCP reporter assay. And to further investigate that, we now made versions of these antibodies where we introduced mutations in the Fc part, either the LALA motif or the LALA-BG motif. With the LALA motif, you basically diminish interactions with Fc receptors. The LALA-BG motif basically leads to also diminished interaction with complement. And this is a way to figure out if Fc-mediated effector functions are really what protect these mice here. And so we made these versions of the antibodies, and the only thing that I'm showing you here is basically here on the left, the wild-type versions of these antibodies have activity, most of them in this ADCC assay. If you have the LALA or the LALA-BG versions, we lose that activity, and the same is true for the ADCP reporter assay. Then when we go in vivo, with some of these antibodies, as you can see here, they are all non-neutralizing, which you see here. We can see that the wild type antibody is protective, but then we either have worse protection or no protection when we go in with the LALA version of the monoclonal antibody or the LALA-BG version of the monoclonal antibody. And so in these cases, we think that the protection is really based on Fc-FcR interactions, mostly probably mediated by ADCC or ADCP. But we also have cases where we don't see that drop, and this might have to do with the amount of antibody that we give, because LALA and LALA-BG have some residual activity, but it might also be that there are other mechanisms at play that we don't understand yet, and so we're still investigating that. We also wanted to look if this holds true in a different animal model, and so we used the hamster model for a subset of these antibodies. We passively transferred antibodies into the hamsters and then challenged them, and we basically see protection, of course, with neutralizing antibodies here in red, so this is in a way the positive control, but also many of the non-neutralizing antibodies can provide protection in the hamster model. This is initial data that we had from a pilot experiment, but we are following up on the hamsters with that now too. So why am I so interested in these non-neutralizing antibodies? When we, again, sorry about the background noise, that's the office next to me. When we started to see the Omicron sequences come out, and big shout out to Tom Peacock here, who really tweeted the first sequence and made a lot of people aware. If you looked at these sequences, you could right away see that almost all of the neutralizing epitopes were affected, right? And you know, what I'm showing you here on the right side is basically neutralizing activity against wild type, beta, and Omicron in different groups. Right here we have convalescent individuals, 2 times Pfizer vaccinated individuals, 3 times Pfizer vaccinated individuals, and here hybrid immune individuals. And what you can see is there is a very steep drop in neutralization, right? So here for the convalescent ones, we go from decent neutralization to wild type to basically non-existent neutralization. And for groups where we start out with higher titers, there's about a 20 to 25-fold drop in neutralizing activity that we observed. If you look at binding antibody, we don't see that, right? We do see a drop, but the drop is about 2 to 2.5-fold. And that's 10-fold less of a drop as compared to the neutralization. And of course, many people have observed that even with Omicron, when you have breakthrough infections, a lot of people might get symptomatic disease, but they are not getting severe disease, right? And so they're protected. And of course, one major factor that might play into that is T-cell-based protection. But we also think that these non-neutralizing but binding antibodies provide a significant amount of protection. And to finish this up, we now started a fun project, and this is all, again, brand new data. When we looked at this cross-reactivity between the variants, we were wondering, what about cross-reactivity beyond SARS-CoV-2, right? What do we see in terms of reactivity to other betacoronavirus spikes, other sarbecovirus spikes, now that everybody has been vaccinated, or even gamma and delta coronavirus spikes? And so what you see is this phylogenetic tree. It spires towards beta coronaviruses with different spikes from the Sarbecos, Heatbecos, Nobecos, Merbecos, Mbecos, and then alpha, gamma, and delta coronaviruses. And we expressed all these spikes and wanted to have a look. And so first we probed these spikes with pre-pandemic serum. That's what you can see here. Here on the very left, and again, these are binding titers. On the very left, you see SARS-CoV-2. Then we have a bunch of variants. Others are Becos, Merbecos, Mbecos, Heatbecos, Nobecos, alpha, delta, and gamma coronas. And what we see pre-pandemic, of course, is reactivity to HKU1, which is clear we get infected with that. OC43, which is also clear. And then cross-reactivity to the bovine coronavirus spike, which makes sense because it's closely related to OC43. And reactivity to 229E and NL63. So all of this makes a lot of sense. When we start to look at people who had a SARS-CoV-2 infection, we see much more coming up. Of course, reactivity to SARS-CoV-2 spike and to some of the variants, but you see a lot of reactivity across, even though it's low, across the sarbecoviruses or even beta coronaviruses. And there is some low reactivity also coming up against delta and gamma coronaviruses. And this actually intensifies when you look at people who had mRNA vaccination or are hybrid immune. So we threw everything together here, but as you can see, you have now really high reactivity across the sarbecoviruses, but you also have substantial reactivity against other beta coronaviruses, and again, some reactivity to gamma and delta coronaviruses. We don't know what this means. We don't know if this is beneficial. We think within the beta coronas, most of this is S2 binding. We think this cross-reactivity that we see to the gammas and deltas might be based on fusion peptide targeting antibodies. We haven't explored that yet, but I think it's an interesting finding. And so just to conclude, the role of human non-neutralizing antibodies is unclear, but some preliminary data from mouse and hamster models suggest they could be beneficial. This could be important in cases when neutralizing activity is low or absent, but spike binding is largely maintained. And these binding non-neutralizing antibodies may not prevent infections, of course, but they could play an important role in protection from severe disease. And we clearly see that the post-pandemic cross-reactivity landscape for coronavirus spikes looks very, very different from the pre-pandemic cross-reactivity landscape. Again, we don't know what that means, but it's something to look at, and maybe there is now more residual protection against new baculoviruses in case we have another pandemic. And here are just my acknowledgements. Of course, a lot of people are involved in these studies. Specifically, Viviana Simon is one of my main collaborators here at Mount Sinai on the clinical side, and most of this work was done by Jordan Clark. And with that, I'll finish, and I'm happy to answer any questions.